Okay, this is prokaryotic cell in under the microscope here, and this is the cartoon. I would suggest you to look at the picture, the cartoon, better because it shows you the structure better. So let's take a look. Okay, we have cell wall. Cell wall is not the most the outermost surface. It's actually in the middle. It's a middle layer. So you have three layers. Okay, one is, let me see. One is cell membrane. Cell membrane is the blue one here. And the cell wall is this red thing here. And then the brown one, this one. So this is the cell wall. And then the brown one is what we call capsule. So cell wall, they are made up of, they don't make, so some of you may have heard that, oh, the plant have cell wall, it's made of cellulose. But this cell wall is different, it's not made of cellulose. So the cell wall is made up of a disaccharide. We call it peptidoglycan. Peptidoglycan, what does it mean? Peptide means peptide chain. Peptide chain, I hope you remember, peptide chain is a connection, is a chain of amino acid basically joined together. And then glycan. What is glycan? Glycan, from the word it means sugar. So it is disaccharide. In other words, it is basically a short polypeptide chain connects to sugar, disaccharide. So that is the structure of the bacteria cell wall. Remember, bacterial cell wall is different from the plant cell wall. Bacterial cell wall is made up of a short protein chain connects to sugar, disaccharide. That's why they said it is unique to bacteria. Protect the bacteria, yes, and keep the shape of the cell. So antibiotics like penicillin interfere with peptidoglycan synthesis, yes. As a matter of fact, a lot of antibiotics, different type of antibiotics, they are interfering the making of protein synthesis in general. They stop the protein synthesis in a cell so that it can kill the cell. So instead of antibiotic, penicillin interfere with peptidoglycan synthesis because there is a protein chain, peptide chain on the disaccharide. So for antibiotics, they may stop the protein synthesis, the making of the peptide chain to put on top of the disaccharide. In this case, then it will like destroying the cell wall, and then the bacteria no longer have the cell wall to protect them. Then it is easy to kill the bacteria. Next one, capsule. Capsule is the outermost the outermost layer of the prokaryotic cell of bacteria. So let's take a look at the structure of the capsule. So the capsule is made of polysaccharide, and it is on the outermost surface of a bacteria. They are hydrophilic. So it helps to prevent desiccation, or they call it dehydration, or losing water from the cytoplasm. They also protect bacteria from phagocytosis. Phagocytosis is you may call it getting swallowed by white blood cells. And then it makes the bacteria more virulent. They are extremely infectious and difficult to kill. It is difficult to kill bacteria because they have a capsule that protects them. And protect bacteria from bacterial virus. Yes, some virus, they can infect bacteria. We call it a bacteriophage. Bacteriophage, they are the virus that will infect bacteria. That also protect the bacteria from that. And last but not least, they have something called fimbria. They are the finger-like structure or extension from the capsule. It helps the bacteria to stick to the surface, stick to the skin on your hand and so on. Flagellum is the structure that allows movement of bacteria. We see that receive flagellum in many bacteria so that they can move around, but in animal cell, in eukaryotic cell, especially in human, try to think about what kind of cell in human being we have flagellum. As a matter of fact, most of the human cell, we don't have flagellum. Well, in the next slides, I'll tell you the answer. Okay, before we start talking about nuclei, I would like to give you the answer. The human cell, the only human cell that has flagellum is sperm. So, sperm. Sperm, when it's singular or plural, it doesn't have S. You don't put an S behind sperm. So both singular and plural is just sperm. And then we talk about nuclei. I already told you what nuclei is. This is the DNA that they say is a location where you can find the bacterial DNA. Yes, the nuclei, they do not have a nuclear membrane to surround the DNA. So the bacterial DNA, they are just, lying inside the cell at a specific region. We call it nuclei. And they are ribosome. I told you that ribosome is a form of organelles. Ribosome is a form of organelles that they make protein. Why is it so important to make protein? Well, you may know that enzymes, they are mostly protein. So enzyme is the one that speed up or catalyze chemical reaction in a cell. So ribosome, they make protein, so they also make enzymes, and that helps the normal functioning of a prokaryotic cell. So bacterial ribosomes, yes, protein synthesis. And then they have two subunits. One is called large, one is called small. And as I told you before, antibiotics usually antibiotic is the one that stops or interferes with the protein synthesis. And some antibiotics, they may target the ribosomes to stop the protein synthesis. And two subunits that differ smaller than the eukaryotic subunit. The size of the ribosome is smaller than the eukaryotic ribosome. In eukaryotic cell, we also have ribosome, but the one that we have, they are larger. So prokaryotic cell, they are structurally simple. Well, when we go to eukaryotic cell, you will see how much more complicated the eukaryotic cells are. Metabolically diverse. They didn't show you this in the previous slides. Bacteria, they have something called plasmid. Let me write it. Plasmid. Plasmid, well, bacteria, they have two types of DNA. One type is in the nuclei. That is the normal DNA that you can find in the nucleus. Another type is called plasmid. Plasmid is like their toolbox. It's like their weapon. They use plasmid to obtain gene from other bacteria. So let's say other bacteria, they are resistant to certain antibiotic, and then this antibiotic resistant bacteria can give out this antibiotic resistant gene to other bacteria. Then other bacteria will receive this antibiotic resistant gene and then put it in the plasmid, so that they will become antibiotic resistant. And that's why it is very diverse, and that with plasmid they can adapt to a lot of different environment, and that's why bacteria, the adaptation, the rate of you may call it mutation, the rate of acquiring new genes so that they can adapt to a new environment is very fast. And that's why we have more and more antibiotic resistant bacteria. Well, let's take a look. Well, the prokaryotic cell and eukaryotic cell. If you look at this picture, you can see that in eukaryotic cell we have a lot more organelles. Eukaryotic cell is very simple. Also, eukaryotic cell is a lot larger than the prokaryotic cell. U means true. Karyo is the same, seed or nucleus. So this type of cell, we have a true nucleus. What do you mean true nucleus? Meaning that nucleus is a form of organelles. It has something called a nuclear membrane. A nuclear membrane that surrounds the DNA. That is called nucleus. Has many organelles. Yes, we will talk about different types of organelles in the next few lectures. And then all eukaryotic cells under the domain eukarya. Eukarya, we have different kingdoms like protista, fungi, animal and plants. Protista or protist. What is protist? Protists, they are also unicellular organisms like amoeba or paramecium. All these are the protists. So fungi, you have mushrooms, animals. Here we have frogs, plants. You know what plant is? Protista. So this one is amoeba. This one is paramecium. I'm not going to spell paramecium because it's a long word. It's not easy to write with my mouse. Amoeba, they do eat paramecium, FYI.